very much for the introduction, Gustavo. Thank you for the invitation to come and speak to you today and to give you a little bit of insight as to why a company like AstraZeneca, why an individual like me might think it's a good idea to put a lot of our proprietary data around our compounds into the public domain so that folks such as yourselves can show us what you can do in terms of modeling and exploring and finding the patterns within this kind of information. So I guess I'll give you a little bit of introduction to drug combinations in general. I know kind of from the speakers we've had over the last couple of days that a lot of you are familiar with the kind of problems we face here, but cancer really is a very heterogeneous disease, especially at the time that we tend to see it with any new compounds or new agents. That tends to be in patients that have gone through several rounds of prior therapy. The cancer has evolved and developed and spread and metastasized, and actually the disease we're working with is a collection of many different diseases within that patient, and we have to figure out the best way of enabling our treatments to affect all of the different diseases that exist. And targeted therapy has had some huge successes. There are patients that respond very dramatically to inhibitors of things like EGFR, etc. But there's a larger number of patients that don't, and those that do tend to go on and develop drug resistance. So the tumors, have ways of bypassing the dependency on that drug target, and really, overcoming the slow of growth that drug might be giving to persist. And there's a hope that going in and targeting multiple components of the signaling within the tumor is the better way of getting a harder hit on that cancer cell, tipping what might be a growth response to one agent into a cell death response to the two agents, and really killing those cancer cells and getting over their ability to then evolve and become resistant. But it's a really complicated problem to try and figure out in which patient the right combination of targets to hit would be to maximize that effect. It's a hard enough problem just with the monotherapy, but when you're talking about every possible combination of different drugs, that problem is exponentially increased. And particularly preclinically, our ability to understand the right targets that will synergize with each other is challenging, and our ability to predict, therefore, which combinations to take through to the clinic is very difficult as well. So, if we think about how combination synergy might manifest itself, and if you think about resistance as the thing that combinations are really trying to overcome, that can come within the cancer cell. So it might be that one drug on its own, isn't hitting the target hard enough, and so the tumor, the signaling from that target is managing to persist through that compound inhibition, and the cell's not quite dying. You're slowing it down. You're not quite killing it, and so actually a double hit on the primary drug target or that primary drug target pathway might be the best way to completely block off that signaling. There's a lot of feedback loops within cell signaling systems as well. If you don't completely block the target, most pathways have a way then of ramping, of feeding back, ramping up that signaling to overcome, the inhibition that you're putting in there, and actually the output of the pathway tends to remain, at a fairly consistent level. The tumors actually, evolve ways of are remarkably smart in the way that they can evolve ways to restore signaling if you don't completely inhibit it. We studied a lot with the MEK inhibitor, selumetinib, AZD6244, which is on the MAP kinase pathway that signals from KRAS to BRAF to MAP kinase, and we were finding cells that would amplify BRAF or amplify KRAS to exactly the right level that the output of MEK phosphor work was restored to the exact level that it was before you inhibited it with the drug. It's really quite remarkable. There can be many ways of driving cancer cell growth. So inhibiting only one of the pathways might not be enough. The cells can ramp up another pathway and completely independently of your drug target continue to grow and continue to persist. There's, combinations also have the potential to work where one drug is inducing a sensitized state that can then be taken advantage of with another. So, we have some great successes here with our DNA damage response inhibitors like olaparib, where, we inhibit the ability of the cells to repair DNA, and then you can go in with things like radiation, to which the cells then have no way of surviving. I mentioned tumor is, cancers are evolving beasts, and so, actually understanding what the different cell populations are within that patient and having, one drug target one, another target another, and, the right sequences of drugs to stop the outgrowth of resistant clones is important. And then obviously, all the press in oncology at the moment is around the microenvironment, particularly the immune system's role in cancer. And so understanding drugs that, where one might be tackling the cancer cell, the other might be boosting the immune response against the cancer or some other component of the microenvironment is important. For this challenge, though, we're working on cancer cell line data, so we're only really able to consider the intracellular routes to resistance. So this is where we're focusing on for this particular challenge. So what was the data? With our compounds, we run high-throughput combination screens where we test, every drug we have in combination with each other in different cancer cell lines to see the effect they have and try and identify which ones seem to have a synergistic effect together. So we had a data set here with around 100 unique compounds, around 900 unique combinations of those compounds in 85 cancer cell lines, which covered 6 different major cancer types and tissue types, and all in all it was well over 10,000 experimental results that we were putting into the public domain. And for each one of those experimental results, we were giving as much information as we could around the compound and what it targeted, and we were giving the full dose-response matrix here. So this is a dose-response matrix where you've got the concentration of drug 1, the concentration of drug 2, and then the map here shows the amount of cell death you get at those different combinations of concentrations of the drugs. And you also get from that the monotherapy response in that cell line as well. So, there's no good having the endpoint unless you have the tools with which to predict that endpoint. So we were, very happy to partner with the Sanger Institute, who released, their large data set for over 1,000 cell lines. But we were thankful they were actually willing to release a bunch of molecular information about those cell lines that wasn't previously in the public domain for the purposes of use in this challenge. And the purpose of the challenge is therefore, can you use all of this data, all of the molecular data, the baseline, before treatment, the baseline data, the baseline information about the cell, about what it is, about its molecular characteristics, to predict which combinations it's going to respond to synergistically, and predict which synergistic compounds to go into which cells with. And why is this difficult? And I guess just something to emphasize to folks in the audience coming from the more computational backgrounds, perhaps, working in cell lines in general, cancer cell lines, it's a problem of multiple N of 1s. Okay, so we have big cell panels out there that are 1,000+ cell lines big. That's not because they represent, 100 or 1,000 of the same tumor type. That's because there's well over 1,000 types of tumor out there, and we've got one cell line representing each one, okay, or 2 cell lines. If you look at some of the biggest success stories recently on the market, so EGFR inhibitors, if you look in the 1,000, the 1,500, 2,000 cell lines that are in GDSC and CCLE combined, there are 2 with the major EGFR driving mutation, 2, okay? So you're not going to find that through statistical power alone, that kind of association. If you look at, olaparib, our DDR inhibitor, and BRCA, BRCA is particularly difficult because the cell lines don't grow very well. And again, there's two breast cancer cell lines in all of these kind of panels that are out there in the world that have these mutations. So, there isn't the statistical power in these data, no matter how big, they appear, to find these associations alone. You need to use knowledge. You need to use clever ways of incorporating pathway information, etc., to augment the statistics to find this kind of information. It's not just a simple machine learning problem. Okay, the context specificity of all of this. So let's pick on the DDR inhibitor, olaparib, as well. So, the way you run these cell line screens, they're typically very short. You run them over 2 days, you throw on the drug, you see what response you get. Okay, but each of these cells replicates at a different rate. So across those 2 days, they've gone through a different number of cell cycles. Drugs like a DNA damage response inhibitor rely on cell cycling. So each cell cycle, the cell picks up more DNA damage, and it's only when it's picked up a catastrophic amount of DNA damage that the cells die. So if you're not actually profiling these cells for long enough to allow this DNA damage to accumulate, you don't see the response to that DNA damage response inhibitor. So you've got to consider the cell cycling times, all these different kind of properties of the cells as well, and the context of the drug and the drug target and what it does, and how relevant the endpoint of response you're measuring is to what that drug is doing, to know that whether and what you're modeling within those cell lines is really important. Don't just treat this as a mathematical problem and assume every drug is the same in here. You've really got to bring the context, specificity into it. You've also got to understand, what are we looking for? So we're not just looking for synergy. Synergy is a difference in response in terms of the way most algorithms out there measure it. What you want is a phenotypic shift. So there's no point in having synergy that takes something that's extremely resistant to something that's mildly resistant. It's still resistant. Okay, so what you're looking for is the thing that takes the resistant cell to something that's sensitive, something clinically meaningful that might elicit something meaningful in the clinic. And so, ultimately, knowledge-aware approaches are absolutely essential for this kind of a problem, for modeling this kind of data. And this is another important thing, right? We need to be able to create things from data that we can realistically then go and measure in a patient. There's no point building a model that needs transcription, methylation, genetics, that needs all of these different data types to work. You'll never get that from a patient. So, we need to build the model that works on the markers we can realistically measure when we actually get to the clinic. Okay, so I'll fly through some of these. I can't see how I'm doing for time, but we'll fly through some of these slides because you all know what DREAM is. You all know why it's important, but why crowdsource? Why crowdsource for us? So, these were data that we generated. We mined them for our purposes, which usually focus on a particular drug. So we don't necessarily take the systematic approach to look for the patterns across these kinds of data. We're interested in one drug and what it combines with, and the stuff that's really active in our portfolio. And so, there's really a huge amount of untapped potential in these kinds of data sets that we're simply not going to look at within for the purposes that we're putting this data to. And so, putting that data out there in the public domain, asking the wisdom of the crowd to tell us what value is in there that we hadn't thought to explore is I think a hugely powerful thing to do, and sharing really does lead to better models and faster. And why DREAM? So we have our own open innovation portal. Why don't we do it ourselves? So you all know these. Gustavo has presented it. But this bottom one is the important one for a company like AstraZeneca. So, and in fact, the whole reason we did this challenge in the first place is Justin and I were designing a one-on-one collaboration with Sage Bionetworks to tackle this kind of a problem. And you think, all right, so we're going to design this one-on-one collaboration, but for the same kind of cost, we can not only get Sage Bionetworks having a go at the problem, we can get all of these other people playing in this data as well and showing us what they can do, and they're the who's who of who we would approach to work with on this kind of a problem as we go. So it just seems like an absolute no-brainer to, rather than, pick off these one-on-one collaborations where, you'll be relying on that person's expertise, the particular modeling approach they're the experts in, you'll be taking, and you'll be ignoring that whole context specificity matters, and you'll be focusing on one way of tackling the problem versus, a challenge where, hopefully you see all the different possible ways of tackling this problem and whether some work well in different contexts over others. And, that's not to belittle I'm sure the collaboration would have been wonderful had we done it one-on-one. But okay, so the challenge with the challenge. So I guess I want to emphasize this bit. This was the hardest bit of the whole challenge, was getting AstraZeneca to approve the release of all of this data. Okay, so there is proprietary information for every single compound that we have in this data set. Okay, or we had at the time in this data set. Each one of those drug projects has their own IP rep. Each one of those drug projects has their own clinical team. I have to convince the IP rep and the clinical team for every single one of these drugs to release the data for their one drug. That putting that data out there in the public domain doesn't compromise what we want to do with it and what they want to do with it. And that is not an easy task. But I want to encourage you guys. There are people like me in pharma who see open innovation as the way the world has to work and the way the future needs to be in the way that we do research. And it's AstraZeneca in particular. I'm really lucky to work at AstraZeneca because our head of research, Mene Pangalos, our head of oncology, Susan Galbraith, are huge champions of open research. And so I had those guys in my corner to usurp it's very easy for a drug project team to just say no. To not even to think there could be a risk, so I'm just going to say no without actually putting the effort in to figure out whether the risk is real or not. And luckily I had those guys in my corner to make them put the effort in and make that critical assessment. But it's not just me. Novartis put their huge PDX screening data out there in the public domain. Merck have also put a smaller combination data set out there in the public domain. We exist in pharma. If you think the kind of data we're producing could be beneficial to this kind of community effort, come and talk to us. Okay, we're not. Don't assume that we're going to be closed to these kinds of opportunities. Choosing the correct measure of success was, I think, the hardest thing to do. I think it's the challenge that I put back to the DREAM community is the one that I don't think is fully solved yet. Because the idea is to be useful, not to be right. And we've really got to kind of push for that. So you've got to really try and think, how do you measure the utility of what participants are producing, and how do you make sure the people giving you the most useful stuff are coming to the top of the rank? The translatable biomarker hypotheses, for example, was the hardest thing we found to try and get out of participants, because the chances are a preclinical model will never make it to the clinic, but it could reveal biology that we can then test and validate. And how do we get that out of participants? Okay, so I'll let Michael talk through what the specific challenges were. I think I've kind of already mentioned that on the previous slide. The incentives that DREAM kind of give to participate. So very quickly, the high-level results and was it successful? So was it successful at attracting the world's talent? Absolutely. So I believe from Gustavo's slide, it still is the fastest recruiting and highest participated DREAM challenge of all time. And thankfully, that wasn't just other pharma coming in to steal our data, because actually people did then go on and submit models and participate. And we got some really great people, including a bunch of companies who were trying to get a lot of money out of us for expensive software participating as well. So it was a really interesting way of seeing how they performed. Did we sample the space and methods? Yeah, we had people using all different combinations of data types. We had people using all different machine learning kind of algorithms. I realize ensemble at the top there is a little bit meaningless. So this is the kind of different ways people were pairing up different machine learning algorithms. There, linear regression being at the core of a lot of them, then paired with some other methods as well. Did we solve the problem of prediction accuracy? Maybe, kind of. We certainly got some really good signal. It wasn't 100% predictive, but some of the top performing models in the cell lines or for the combinations that they perform well were certainly predicting within the kind of margin of variability you get within the data. Anyway, but only doing that for a subset of the combinations or the cell lines, and I think that's an important thing, because a lot of the winning teams actually predicted the same subset of combinations or the same subset of cell lines. So actually there's a big old gap within this data that no one's predicting well. There's certain drugs, certain cell lines that no one has managed to build models to predict well on, and this has been the focus of some of our bonus challenges. Who can predict that black space? Who can figure out why? Is it because we don't have the right data to model the biology for those cases, or is it because the algorithms that have been used actually don't suit the shapes that those data show for those particular cases? And if we focus on them, can we do better? And did we solve the problem of informative biomarker insight? No, I don't think we did. So this was a very difficult thing to do. How do you incentivize this? How do you get people to reveal the kind of the feature associations, the biomarkers that are coming through from their predictions, because it's a lot of work. Usually this isn't something that's automatically reeled out of the end of most people's algorithms. It's a lot of work to kind of build this and substantiate this. Some teams did set an example. So, some teams gave us really clear methods that we could go and reproduce ourselves to get that data out. Some had algorithms that did give out, really nice kind of results and so on, and some kind of went on and built up some of those hypotheses, but the majority didn't. The majority, it gave us biomarkers for less than 10%. They just ticked the box that they needed to meet the minimum requirements for submission. And then a lot kind of, the algorithms, we can't even really run them ourselves to get that biomarker insight out either. So, there's some value in there. We're working on it. This is the bit that we're really spending the time on now to get the value out for the paper, to understand the biology that's been revealed here. But it's something that you guys really need to think about more, is how do we incorporate this as one of the primary success measures of these kinds of challenges. It's a very hard thing to do non-subjectively, but it's the most important thing we can ever get out of these challenges, I think. So, in conclusion, crowdsourcing does work really well if managed really well, and if you have a fantastic team of people with the kind of energy it takes to push them forward. We were right, the problem is difficult, but it's popular. We got a lot of participants. I'll let Michael talk through, the secrets to success and how the different algorithms perform well, and indeed the winning teams themselves as they come up and present. The well-performing teams made overall similar predictions. So, how can we stimulate more diversity in the way people are looking at these kinds of problems? It's not, thinking about ways where we can reward people who predict well within subsets of the data, not necessarily only those that have, the overall kind of better prediction. And how do we better reward that focus on translatability and utility? So a huge team of people involved. The biggest thanks goes to the participants. Obviously, I think one of the things I was most pleasantly surprised with was, looking across the discussion forum throughout the challenge, was how willing and keen and proactive all the different teams were to help each other out. I thought it was absolutely phenomenal, how seeing real crowdsourcing in action. It wasn't just a challenge where everyone was competing with each other. It really was a community of people trying to help each other solve a problem. And obviously, the key people involved in this, the best description of how long this took was when we started. Michael Mendon over there was a PhD student at the EBI. During the middle of the challenge, he was then a postdoc over at Aachen, and he's now a senior scientist at AstraZeneca. So, that's how long these challenges kind of take. Someone can go through 3 careers during the process of the challenge. And you'll see this slide again, and I'll leave you on this one and wrap up there. Thank you very much. Do we have time for questions? Or are we?